morning. Buenos días a todos. Bienvenidos. Welcome to the third day of our joint symposium. We will start immediately with the next session. This will be first Alejandro Bravo del Biotechnology Institute of the University of the UNAM. And later will speak David Heckel, director of the Max Planck Institute for Chemical Ecology in Jena. And the moderator will be now from Francisco Javier Figueroa Martinez, de la Metropolitan Autonomous University. So I would like to invite the speakers to come to the podium and that Alejandra Bravo empieza.
We are ready? So, wait a minute. Good morning, everybody. Welcome to the third journey of this symposium, Frontiers of Science, hosted by CONACYT and the Max Planck Society. This morning, we are starting with the session Chemical Ecology and Plant Sciences, and the first two speakers are both remarkable researchers. First, we will have Alejandra Bravo. She got her PhD in biomedical research at the National University of Mexico, UNAM, worked at UNAM since 1989, now is professor at Institute of Biotechnology at UNAM too. She has been level 3 member of the Mexican National Researcher System from 2004 to date and has over 160 scientific publications. Her main line of research is the study of insecticidal proteins produced by bacteria. Next, we will have David Heckel. David Heckel was awarded the PhD in biological science from Stanford University. He was named director at the Max Planck Institute for Chemical Ecology in Jena, where he founded the Department of Entomology. His research interests include adaptation of generalist versus specialist herbivorous insects to chemical defenses of their host plants, resistance to chemical and biological insecticides, the genetic basis of host shift, mechanism of sympatric speciations, amongst many others. So I need to tell you the dynamic of the session, and it will be as follows. First, we will listen both speakers. Each of them will have 20 minutes for their talk, and then at the end, we will have the question and answers sessions. If you have any questions, please write it down and hand them out to CONACYT staff, and I will read them at the end of the session. So please, Dr. Alejandra Bravo, welcome to this symposium. It's your turn.
Hello, good morning, everyone. It's an honor to be here. I thank the organizers for the invitation to come and give this presentation. Well, the work that as you heard, I will talk about the Bacillus thuringiensis toxins that are used for insect control. Okay, insects are very important because they are responsible of crop losses. 20 to 30% of the crops are lost only due to insect attack. And so the growers had to control the insects, and they use chemical insecticides. Although these chemical insecticides are very toxic to humans, have been used for many years, and the insects have become resistant to this type of chemical insecticides. So the solution for the growers is to increase the doses of the chemical insecticides. So what is the alternative? One alternative is Bacillus thuringiensis insecticidal proteins. This is a bacteria that during sporulation produce huge quantities of some kind of proteins that we call cry because they make crystals in the mother cell compartment. So this is the spore. and this is the crystal produced by the bacteria. And this crystal is composed of insecticidal proteins. These proteins are highly specific against the insects, are not toxic to humans, and are biodegradable. The larvae ingest the crystals, and then the crystals are solubilized in the mid-good conditions of the larvae that are very high pH. This is maybe is the biological system with the highest pH. Can be 10 to 1030 pH. We have many compartments in the cells that have very acidic pH, but very alkaline pH is very weird. And we can find this alkaline pH in the midgut of the Lepidopteran larvae. The crystal is solubilized, and then the individual proteins are activated, and they are able to bind to the receptors that are located in the midgut microvilli. In minutes, the toxin makes pores in these cells, and the cells becomes to be broken, and the spores that are there can be germinated, or other bacteria can germinate there because it's a nutritious medium, and in few days the larvae die. So this is the larvae that we work in the laboratory, Manduca sexta. Is a pest of tobacco. It's not very important as a pest, but it's a very good model for biological research. And this is a healthy larvae, and this is a dead larvae after eating BT. The cryotoxins are used as insecticidal sprays. For many years has been used, more than 58 years has been used as sprays, but also has been used in transgenic plants. The crygene has been introduced into the genome of the plant. Now the plant can produce these proteins. When the larvae come and try to eat this plant, it is fed with a toxin, and so the plants do not require to be sprayed with any crystal, any chemical insecticidal, and there are no reports of human intoxication in hospitals. There are not effects in animals in farms. There are not environmental damage because this is a protein and can be degraded very fast in the environment. The main problem could be the selection of insects that become resistant to these toxins. This figure shows one group of proteins. These are the three-domain toxins, and the three-domain toxins are the most studied proteins produced by this bacteria, and as you can see, we have quite a big number of them. At least we have 55 different subgroups of the three-domain toxins, and we have already 9 structures that have been solved. As you can see here, these proteins are composed of 3 domains, as the name said. Domain 1 is the one that is inserted into the cells to make the pore, and domain 2 and 3 are involved in receptor recognition. The structures are very similar. Some toxins are able to kill Lepidopteran larvae, others kill Dipteran larvae or Coleopteran larvae, and we have some toxins that are able to kill nematodes. You can see here that they have this three-dimensional fold, even though that they share very little identity. Each toxin kills a limited group of insects. What do I mean with this is that here I put different cryotoxins, cry-1 toxins. All of them have more than 45% identity, and different Lepidopteran larvae. And as you can see, there is not a single protein that can kill all the Lepidopteran larvae. So each toxin can kill only a very limited number of insects. And that's very good in the ecological point of view, because you have the poison to kill only the target that you want to kill. So in other words, other insects are protected, and they will not be susceptible to these proteins. For instance, bees or any good insects that could be beneficial for the environment would not be susceptible to this type of proteins. The mechanisms of action of these toxins at the cellular level, if we feed the larvae with the crystals, and we look inside the cell, inside the larvae, what is happening. We can see that the larvae, this is the midgut tissue, and the midgut tissue is a very simple tissue. It's a monolayer of cells. Inside this monolayer is the lumen, and in the lumen is the food. And outside of this monolayer is the hemolymph, that is the blood of the insect. So if we localize where the toxin is located, here is with antibodies, we localize that the toxin is located in the microvilli. All receptors are located in that microvilli. Few hours later, if we analyze what's happening, the toxin inserts into the membrane, make a pore, and kill the midgut cells. And the cells are destroyed. As you can see here how the tissue was damaged. The cells become swelling until they break down. And it's because the toxin is making a pore, water and ions are going through, and, but the toxins remains bind to the microvilli all the time. If we look at the molecular level, what is the mechanism? The crystals are ingested, and as I mentioned, the conditions in the midgut are adequate to solubilization of the crystals. Now the soluble protein can be activated by proteases that are present in the midgut, and now the toxin is able to bind to these two receptors that are highly abundant in the membrane, alkaline phosphatase and aminopeptidase. We have identified the epitopes involved in this interaction. This is a low affinity interaction, but this helps to have the toxin close to the membrane. And now the toxin is able to bind to another receptor, the cadherin, that is much less abundant, at least 500 times less abundant in the membrane. And this interaction is a more complex interaction involving 3 different epitopes in the toxin that binds to 3 different epitopes in the cadherin. This triggers a conformational change in the toxin and allows an extra cleavage here. Alpha helix 1 is cleaved out, and this triggers oligomerization of the protein. Now the oligomer is able to bind with high affinity to these 2 receptors, alkaline phosphatase and aminopeptidase, and that's what it is called ping-pong mechanisms. And this interaction is now with high affinity, and the function of these receptors is now to induce the pore formation and the membrane insertion into, of the toxin that will kill the larvae. We know that helix alpha 3 is involved in oligomerization, and helix alpha 4 is involved in membrane insertion. Recently, we found that not only the activated toxin is able to bind to the cadherin receptor, also the protoxin was able to bind to the cadherin. So here I show a binding assay showing that we purified a fragment of the cadherin that contains the 3 epitopes that binds the toxin. And you can see here that the binding of the activated toxin is very similar to the complete protoxin. The affinity is 3 nanomolar versus 6 nanomolar is quite similar. So what's the meaning of this binding? So in the mode of action and I mentioned is the protoxin. This is the structure of the complete protoxin. It has to be activated, so the activated toxins can bind to receptors. But now we are seeing that the protoxin is able to bind to the cadherin receptor. So what's the meaning of this interaction? We analyze these interactions. We incubate, for instance, trypsin-activated toxin with cadherin, purified cadherin and midgut juice from the larvae, and half an hour, then we incubate like 3 minutes at 25 degrees, 50 degrees, or 100 degrees, and resolve the molecules in a SDS phase that was revealed by Western blot with antibodies. So we can see that during this incubation, the toxin makes an oligomer from 60 kilodaltons, makes an oligomer of 250 kilodaltons, high molecular weight protein. But this oligomer is quite sensitive to heat and can be destroyed if the samples were heated at 50 degrees or 100 degrees. If we make the same experiments with the protoxin and incubate the protoxin with cadherin and midgut juice, the figure is totally different. A different oligomer is formed. This oligomer is more resistant to heat, so that means that the conformation of the oligomer should be different. And if we analyze the pore-forming activity of these oligomers in black lipid bilayers, sorry, I don't know. If we analyze the pore-forming activity, these are a black lipid bilayer there are 2 compartments, and the black lipid bilayer is in the middle. So if one pore is inserted in between these 2 compartments, we can measure the transfer of ions through the membrane. So if we saw here the pores that are formed by the protoxin after incubation with the cadherin and the midgut juice, you can see this is the pore is closed and then opens to a single stage and remains open all the time. So it's a very stable pore-forming activity with a single conductances, and this is the typical current voltage plot. If we analyze the same with this oligomeric structure, you can see here that the pore is close, open, close, open, close, open, and the opening is not as a single conductance like here. There are multiple conductances stages, so this is more variable pore-forming activity. There are two oligomers, and there are two different pores. So we came to this conclusion that there are two different pathways, one from the activated toxin and one from the protoxin, making two different pores that have different characteristics. This is more resistant to temperature, is more efficient to insert in synthetic liposomes, and it uses pores with very stable pore-forming activity and high upper probability. In contrast with this pore that is highly heat-sensitive, requires receptors for insertion, and induce a pore with multiple conductances. Two different mechanisms of actions could be triggered. So are those mechanisms actually working in vivo? So to prove this, if these were two independent mechanisms, insect populations that become resistant to activated toxins, for instance, if we have an insect that becomes resistant to the activated toxins, maybe they will be still susceptible to the protoxin if these were two different mechanisms. So we make contact with different researchers in the world that have different populations of insects that have been selected with activated toxins. So all these are populations. There are 7 populations, Diatrea saccharalis, Helicoverpa armigera, or Helicoverpa cia larvae, that are resistant to activated toxins. So you can see here that activated toxins in the darker bars. These populations are 200, 500, 1,000 or 5,000-fold higher resistant to the activated toxins. When they were fed with the protoxins, all of them were susceptible to the protoxin. So this proves that there are two mechanisms different, triggered by the activated toxin or by the protoxin. And both pathways are involved in toxicity. The main problem with this type of proteins, as I mentioned, is that the insects can develop resistance. And actually, we have already different examples all over the world. These are the different countries where resistant populations have been isolated in the fields, not in laboratory conditions they have been selected, but also in the fields to BT corn or to BT cotton. Different insects have been developed already resistance in the fields, or to sprays in Canada and in Hawaii in greenhouses. So the future is that more insects will be resistant to this type of proteins. What are the mechanisms involved in resistance? So any step in this mechanism of action, if it is blocked, the result will be that the insect becomes resistant. So for instance, if the toxin activation, because less proteases are produced, then less activated toxin, then the insect becomes resistant. So, however, insects that are affected in proteases, they are also affected in digestion, and they are not very healthy, and this type of insects has been isolated only in laboratory conditions. Also, insects that have enhanced immune response can be more tolerant to the toxins, but this type of resistance mechanisms do not result in very high levels of resistance. Other type of resistance is receptor shedding of arterial lipid rash, but these also are affecting the biology of the larvae and in a very important way, so the insects are not very healthy and cannot survive in the environment. The most common mechanisms of resistance are mutations that are located in the receptors, alkaline phosphatase, aminopeptidase, or cadherin, or mutations that lower the expression of these receptors. So these are all the examples that we have in the literature that are located in this type of receptors. And in the last 5 years, a new mechanism of resistance was reported. It's a new component. Mutations in ABCC2 transporter are linked to very high levels of resistance. And the ABCC2 transporter mutations here has been reported in a number of insects already. They are proteins that are engaged in the import and export of different molecules across the membrane, like xenobiotics, metals, and insecticides. The mutations here result in a very high levels of resistance, and we think that this molecule is involved in oligomerization and membrane insertion, but I am not talking too much about this protein and the work that we have done with the ABCC2 transporter, because we have here the expert in ABCC2 transporter, this is Dr. David Heckel, and he will talk about the role of this protein in the mechanism of action of Bt toxins. So I want to talk just to end with about other other projects that we have in the lab. We know that the cadherin protein is involved in induced oligomerization of the protein because it helps to the helix alpha 1 to be cleaved out. So we have different here mutations in the cadherin. Like here, this is a transporter insertion that blocks the transcription of this gene, or other mutations that also make stop codons or deletions in the genes that make shift frames. So mutations in the cadherin genes do not produce the cadherin, the protein is not able to make oligomers and is not toxic. So we argue, what's happening if we make a cryotoxin, a modified cryotoxin that lacks alpha helix 1? If we make this construction, then when the toxins came into the midgut, it will be able to make oligomers even in the absence of the cadherin, and these oligomers will be able to bind to the receptors, the following receptors, and make the pores. So we made this type of constructions, and you can see here that the protein, the wild-type protein, when it is activated with trypsin, gives a 60 kilodalton protein, and in the presence of cadherin fragment that binds the toxin, produce the oligomer, and our mutants that we call CRY1AB mode or CRY1AC mode, they are able to make oligomers even in the absence of any cadherin fragment. And these oligomers, we later demonstrate that if we increase the pH and add lipids, we can see that there is a very high amount of oligomers in the CRY mode toxins and not in the CRY1AB toxin, because they will be formed only in the presence of the cadherin. So we test these proteins against resistant insects. We make contact with Bruce Tabachnik from the University of Arizona. He has a population of pinball worm that is a pest in cotton that has mutations linked to the cadherin gene. These are the resistant line and the susceptible line. You can see here a big difference, more than 100,000-fold resistance. When we send our proteins to him and he tested, he found that our proteins are able to kill the resistant line. So we then make contact with many other researchers all over the world that have different populations of larvae that were affected in different receptors, in cadherin or ABCC2 or in aminopeptidase. All these populations were kept by these researchers. We make contact with them and we send our proteins to them. So they test our proteins and these are the results. So this is the resistance ratio, and as you can see here, all these larvae are highly resistant to CRY1AB or CRY1AC toxins, but when they were tested with the CRY1AB mode, they were, the resistance was overcome. You can see here, these are different resistant lines and affected in different receptors. Our hypothesis was that our mutants will be able to block the resistance of the cadherin and overpass this cadherin resistance, but maybe not all the others. And to our surprise, these mutations were able to kill all resistant larvae affected in all different receptors, suggesting that oligomerization and membrane insertion is very important in all cases. So CRY1AB toxins are able to kill insects that have developed multiple mechanisms of resistance affected in cadherin, but also in ABCC2 transporters. So these proteins are very important tools to control resistance development in the field. And with that, I finish my presentation, and thanks for your attention.
Thank you very much, Doctor. Now we will have Dr. Heckel with his presentation.
Thank you for the introduction and for the invitation to speak here. I'm a foreign member of the Max Planck Society since 2004, and I feel incredibly honored and privileged to be a scientific member in a society that is unique in the world and giving its directors complete freedom to pursue any aspect of research that they would like to do. And we can see the fruits of this in this incredibly diverse symposium today. So I'm absolutely thrilled to be here. I'm also very lucky because the previous speaker has completely introduced the topic to you, so I can jump right into my contribution. Unlike most other groups that have looked at the interaction from a biochemical point of view, identifying the proteins and the types of interactions involved with the toxin, I've taken a different approach, and that is a genetic approach to analyzing Bt resistance. So starting with resistant strains selected by ourselves or by our colleagues, across them to susceptible strains, create a genetic linkage map with markers, and then look for a correlation between certain of these markers and the resistance in progeny of these crosses. And so this is a standard approach that's been used for plant genetics, crop genetics for a long time, but not quite so well used in insect genetics, except, of course, for Drosophila. And so this is useful because this can confirm candidate genes. If we know a gene, for example, cadherin, maps is very close genetically to a marker that's associated with resistance, that suggests that the cadherin is in fact involved. It's also very important in rejecting candidates because we have many strains where the cadherin is not genetically linked to the resistance. And so we might have to take another approach. And we were confronted with this problem in working with a strain that we had been working with for many years. This strain had a mutation in the cadherin gene that conferred very high levels of resistance. This was in the cotton pest Heliothis fluorescens, a pest of cotton in North and South America, that has developed resistance to all chemical insecticides and for which the Bt protein is a good source of control. When we selected this strain over many generations in the lab with toxin, we found that the resistance level increased even higher, leading us to suspect that there was another genetic change in this strain. And this was complicated because now there were two resistance genes in the strain, and we had to sort of separate them out in order to do the linkage mapping. We expected one of these proteins that Dr. Bravo has told us about, aminopeptidases, alkaline phosphatases, and other ones, would be linked to the resistance, but we found that none of them were linked to the resistance. And so we were left with the alternative that we had to identify this gene by positional cloning. And this was rather difficult because when we embarked on this project, we had no genome sequence. We had a very crude genome map. The model Lepidopteran insect, the domesticated silkworm, sequencing was in progress. They had a genetic linkage map, but still no real good sequence. So we had to jump back and forth between the genetic linkage map of the silkworm chromosome 15 and the chromosome to which we had mapped the Bt resistance, starting at one of the chromosomes, getting closer and closer to the region where the markers were tightly linked to the resistance. We finally ended up sequencing genes in this region, and we found a mutation involving 20 base pair deletion in a protein that completely inactivated the protein by truncating it at amino acid 99. What was this protein? This was a real surprise to us. This protein was a member of the ABC transporter superfamily. They have a membrane structure that looks like this. They have 12 transmembrane membrane domains that stick in the plasma membrane, 2 internal domains that bind to nucleotides, and the three-dimensional structure looks something like this. So this is the outside of the cell, this is the inside of the cell, and this was what our linkage mapping, our positional cloning came up with. This was a total surprise because no study at all had ever identified any interaction between any cry protein, three-domain cry protein, and an ABC protein. So we had to figure out what was going on. And so one powerful technique for looking at the interaction between proteins and Bt toxins is to actually label the toxin with a radioactive label or a biotin label, and then incubate them with little vesicles. And so the brush border villi of the membranes, if those are sheared, those will bleb up into little vesicles, expressing the receptors on the outside. When those are incubated by labeled toxin and then centrifuged down, if there are receptors on the surface of these vesicles, the toxin will stick to them, and then you will be able to recover them when you take the precipitate and run that out on a gel. On the other hand, if the vesicles do not have receptors for the toxin, you will not recover the labeled toxin in the gel. And we found that the mutation of this ABC protein and the mutation of the cadherin drastically affected the binding of the toxin to these membrane receptors. And so the normal wild-type strain bound to 3 variants of the 3-domain protein that are especially toxic to these insects, Cry1A, Cry1AB, and Cry1AC. The original cadherin knockout mutation enigmatically did not bind to this one, but did bind to these 2, and we couldn't understand that because the insects were actually resistant these 2, but the membrane vesicles actually bound to them. Our ABCC2 knockout strain had the complementary pattern. They bound Cry1A, but not Cry1AB and Cry1AC, and the double knockout strain no longer bound any of these toxins and was highly resistant to all 3 of them. So something was going on with binding in this strain, but what could it be? And so I had to come up with an explanation, so I just went out on a limb and looked at the actual way that the ABC transporters actually transport. How do they transport small molecules from one side of the lipid bilayer from another? These are molecules that cannot diffuse through the lipid bilayer because they're not lipophilic, they're charged, highly polar, but the way the ABC protein transports them across follows a cycle. So this is the outside of the membrane on the top, the inside of the membrane on the bottom. At the beginning, the conformation of these transmembrane domains is actually closed to the outside surface of the membrane. And the small molecule that's waiting to be transported is sitting in the pocket that's facing the inside of the membrane, or it's actually in the lipid bilayer facing the membrane. So what happens is that these 2 nucleotide binding domains bind to ATP, adenosine triphosphate, one of the energy currencies of the cell. This binding causes a tremendous conformational change, bringing these 2 domains together, forming a dimer, and then changing the conformation of the transmembrane domains, kicking the small molecule out of the cell. The ATP is hydrolyzed, the ADP is released, and the membrane protein returns to its original conformation. My hypothesis is that in this opening and closing, there's a moment by which the toxin preport structure that Dr. Bravo has told us about can actually insert and then be pulled into the lipid bilayer. And so this is shown, these hydrophobic lipid patches in the mouse model of the ABCB1 protein are exposed to the surface transiently. So this was the model. This actually would explain the actual last step, the insertion of the pore into the lipid bilayer and eventually killing the cell. So, well, that's all very well and good. So we started to look, and other people started to look at other species. And the next paper to be published was in the domesticated silkworm Bombyx mori, where among the 200 or so cultivated strains in the stock center, they found some that were highly resistant to Cry1AB, and they can do positional cloning even better than we can in Bombyx. They found a mutation. Actually, they found several amino acid substitutions in the member of the same family. But what was enigmatic was that there was a single tyrosine insertion in one of these tiny extracellular loops. And by combining all these different substitutions with one another, and doing germline transformation, which they can do in bombyx. They can actually take a gene from one strain and stick it into the genome of the other, which we cannot do yet in our insects. They found that this single tyrosine insertion showed that that would confer resistance to Cry1AB, a very small change in this protein. Most of the other species that have given examples of mutated ABC proteins are much more drastic, like we found in Heliothrus fluorescens. So in another cotton pest, the old world cotton bollworm, Helicoverpa armigera, a splicing defect between the introns and the exons led to a premature stop codon, completely removing the second nucleotide binding domain. This confers resistance to Cry1AC. In the diamondback moth, which is a worldwide pest of cabbage and related vegetables all over the tropics and the subtropics. There is also a knockout known, but in a strain in China, it was found that there was no obvious mutation in the ABC protein. However, the expression level was lower, and knocking these down with RNAi could make the insects survive BT toxin better. And it was found that there was actually a MAP4 kinase mapping very close to the ABC protein that affected the expression level of that protein. And RNAi against the MAP4 kinase can actually increase the larval mortality because it increased the amount of expression of the ABC protein. So here's the expression level that's being, when that's tuned down, the insects can tolerate the protein better. Mapping studies in the European corn borer, a very important pest of maize, against which the transgenic maize has been developed in the US, show by mapping that the almost total resistance in the resistant strain maps very, very close to an ABC protein, but they don't know yet what the mutation is. And a slightly different toxin, the Cry1C toxin, that's more active against the Spodoptera family, these are the armyworms, is also associated with knockouts that completely also remove the second nucleotide binding domain. Now, these are all one particular type of ABC protein among the 80 or so present in the insect genome against one particular type or closely related family of ABC, of BT proteins that have been in cotton and corn and target very specifically those species that were shown in the graph by Dr. Bravo. So the unique things about these, about how specific they are. And so that's why we were very, very surprised when we looked at the beetle-active toxin. Chrysomela tremula, a leaf beetle that eats poplars, was found resistant populations were collected from transgenic poplar that the French INRA lab had created. And we found, my postdoc found that in a completely different member of the ABC protein family, ABCB1, this conferred resistance to the beetle-active toxin Cry3. So the beetle-active toxin will not kill the Lepidoptera. Mutations in the beetle-active toxin have no effect on the Lepidopteran toxin, but we now have a new relationship between another BT toxin and another ABC protein. And to carry this theme on, yet another member of the ABC superfamily, ABCA1, was found to be mutated in multiple places, conferring resistance against the Cry2AB protein, which is the second protein that's been put in the cotton to protect it against insect damage. And so what's really amazing, and nobody really could have anticipated, is that the sequence similarity among these toxins is on the order of 30%. The sequence similarity among the different ABC proteins is on the order of 25 or 30%. Yet there's a relationship between a BT protein toxin and an ABC protein in every case. Could this be a truly general mechanism of the mode of action of these three-domain crytoxins? Well, we need to come back to what's going on with the cadherin. And the most powerful system that's actually been used to address, there's actually a synergistic interaction between the ABC protein and the cadherin, is a system that's in wide use for studying ion channels, but has only recently been applied to study the BT pore, which itself actually passes cations. This uses the gigantic oocytes, or the egg cells, of the clawed frog, Xenopus, These can be isolated from the female, injected with messenger RNA. After a few days, the protein will be expressed in those single cells, and if this protein is an ion channel, then you can stick electrodes to it, apply a constant voltage across the membrane, and then measure the current flow. So this is a very precise way of characterizing the pore. And so the main advantage is that, first of all, we don't have any other insect proteins involved except for the ones that we've injected. And second of all, we can do quantitative measurements of the current flow that can give us precise effects on the pore formation. And so the results of this experiment of this group in Japan, we haven't done this ourselves, is that when the ABCC2 protein alone is injected in there, this promotes very efficient oligomerization of the toxin. So we get tetramers forming out there, and pore formation and current flow. If the cadherin alone is transformed, there is oligomerization, abundant oligomerization, but there's pore formation, but with 6,000 times less current flow. But when both are transformed into the same cell, there's a 7-fold increase of current flow, additionally present by the cadherin, caused by the cadherin. So there's definitely a synergistic interaction, and we don't really understand what's behind this interaction. But this really does show that it's the ABC protein that's truly key to most of the pore formation that must be killing the insect. If this is a general mode of action, are there general anti-resistance strategies? Can we actually use this across many different types of insect resistances? Now, this might give a clue. I mean, we have to ask ourselves, what are those ABC transporters there for in the first place? I mean, presumably they are protecting the insects by exporting toxic substances from inside of the cell to the outside of the cell so the insects can get rid of them. And there are many cases in human biology known where drug resistance to cancer chemotherapeutics is caused by an increase in pumping out of the tumor cells by an increased expression of ABC proteins. And so presumably it was adapted to those cells to have those ABC proteins. Maybe knocking them out can confer a cost that we can exploit. And in fact, another group has already started looking at this. This is a very important class of chemical insecticides, a polycyclic lactone that was isolated from Streptomyces bacteria. This discovery won the Nobel Prize because these are incredibly effective insecticides and also nematicides. They target insects and nematodes and worms, helminthic worms, because these two types of organisms have a chloride channel, particular type of channel in the nervous system that we mammals do not have. So they're actually very, very safe to mammals, and in fact they're used as anti-helminthics. When your dog has worms, you feed them one of these compounds, it clears the worms. The dog actually has ABC proteins in the blood-brain barrier. Actually, collie dogs do not, and so collie dogs are actually very susceptible to these. So here's a case in which ABC proteins are now known to protect mammals against the toxic effects of these proteins. So in group of China, very cleverly started to test BT-resistant strains, and they actually found that a strain that was highly resistant to Cry1AC due to a knockout of the ABC protein was 9 times more sensitive to abamectin. That is, BT resistance was counteracted by increased susceptibility to this insecticide. And Dr. Bravo, in collaboration with him, found that those cells actually, the BT-resistant knockout accumulated more of this toxin in, and they couldn't export them quite so efficiently. So this sounds very promising, but we wanted to know whether this would work in our species. And so we're taking actually a general approach to look at all of the compounds that these ABC proteins might be transporting, which we can do both with living larvae, since we have the knockout strains, but also with membrane vesicles. So if you make inside-out membrane vesicles, the ABC proteins will be pumping the stuff from the outside and the inside. If you incubate with various bodily fluids or various chemicals and then do a differential screen, you can possibly find transport substrates of the ABC proteins. Or if you collect the excreta or the frass of the resistant and susceptible. So we're also looking at differences on this level. And we found, in fact, that the ABC protein of this species does differentially transport one of the two isomers of avermectin. Some of it gets through the cells anyway, but some of it is transported. But when we actually did bioassays with avermectin, we found that there was a problem. It's too potent, it works too well. If you apply avermectin on the outside of the insect, well, the ABC protein is in the midgut and it doesn't protect. If you feed the avermectin to the insect, it eats and eats and eats until it can eat no more. It stops eating because avermectin paralyzes the insect. It paralyzes the chewing muscles of the insect. It paralyzes the body muscular of the insect, so it just sits there. It paralyzes the peristaltic action of the midgut, so it can no longer excrete, but it doesn't kill the insect. The heart keeps beating, and you can watch the heart beat for 5 more days before the insect finally dies. The heart is on a different cycle. It's an autonomously stimulating thing. And so the problem is, no matter how much avermectin you give, the insect will eat exactly as much until it can't eat anymore. So the ABC protein really cannot do any protection. So unfortunately, that is not working. Now let's look at the plants that these insects are eating. And there's a lot of work in our insect with one particular class of compounds present in the mustard family, the brassica family. This is the active ingredient in mustard. The thing that gives us tangy tests when we eat mustard are actually this plant compound that's degraded by an enzyme in the plant, releasing a number of toxic compounds. Insects have evolved ways to detoxify these by conjugating them to glutathione and metabolizing the products. And so in working with this group, that we found that the BT-resistant insects actually do a worse job of exporting some of these compounds. So there could be some relationship, but this only works for insects that are eating cabbage. Our insects eat cotton and also maize. So we're keeping on looking to try to find out what the natural substrates are. And we know we have a lot of work to do because this is the closest human homologue. There's a huge list of hundreds of transport substrates, including xenobiotics, but also naturally occurring compounds. And so, unfortunately, avermectin is not a good candidate, but we're looking for other things. We want something that's not a pesticide. It's not toxic. All it does is interfere with the process that the insect has evolved to get rid of it. And so those resistant insects should be especially sensitive to this normally non-toxic system. I want to leave you with the fact that we have a tremendous diversity of BT toxins, more than 300 different known, with an incredible diversity of different structures. They have all the same 3 domains, but the protein sequences are highly divergent. We also have hundreds of different ABC proteins in insects, and I believe this is a truly ancient interaction. These ABC proteins have diverged from one another 1 billion years ago. I mean, these are found in bacteria, in absolutely everything. Could the interaction between these prokaryotic parasites and the eukaryotics be one factor that's caused this incredible diversity? So this is a very topical topic. BT resistance is growing. We're taking a genetic approach to try to understand the fundamental mechanisms in hopes that we can actually do something about that. So thanks for your attention.
If you have any questions, please pass it down. The first question that we have is, do you have 2 microphones in there if you need to talk? There is a question for Alejandra Bravo. Are there other strategies known for the other insect resistance mechanisms? For example, receptor shading, toxin alteration, etc.?
Well, other strategies that to cope with the insect resistance, the best strategy is try to find a new toxin that can bind to a different receptor. And although it's not an easy task, has been new toxins, different toxins has been described in the literature. And also other strategies that have been used is to use a complete different strategy, is to use RNAi, silencing with RNAi, double-stranded feeding the larvae that can block specific genes of the larvae that are very important for the development. That can could be another strategy to kill the resistant insects. So it's like a different transgenic plants that can be developed with different strategies that will not that will be able to kill the already resistant insects. No? Right now, we have different toxins like the BIP3 that has a very different mechanism of action from the CONACYT toxins, and other proteins that are produced by different bacteria are now also being studied to be transformed in plants, and this RNAi could be a different strategy also.
Okay, thank you. I have another question for Dr. Bravo. The cryo system work also in arachnids? In arachnidos?
No, the cryotoxins do not kill arachnids. No, it's only Lepidopteran, Dipteran, and Coleopteran larvae. Hymenopteran, we have some toxins that can kill Hymenopteran. I don't remember if there are other orders. Nematodes.
And so some nematodes can be killed by BT toxins. And actually, Raphael Royan has taken one of these toxins and actually trying to develop it into something that can be taken by mouth to cure the worm that causes river blindness. And so this was originally discovered by a company, Mycogen, that were interested in controlling plant parasitic nematodes, but they're actually active against animal parasites as well. And so it looks like a very, very promising, because once again, it's completely non-toxic to the humans.
Good. Any other question from the audience? I have a question for Dr. Heckel. Could you talk a little bit more about coevolution between ABC transporters and cryotoxins? Just there is any link already proved or something like that?
The only proof we have is species comparisons. What would be better proof is we could find different populations of the same species with changes in the amino acid sequence that correlated with the resistance of those species. So in much of my other work, I can find evidence like that when I study closely related populations of the same species and find different levels of adaptation. We haven't found that level of evidence yet for BT, though. But I do believe this is what's going on, because it just seems no other way to explain the fact that this bacterium over coevolution has basically targeting the same target. I don't think this is an independent re-evolution of hitting the ABC protein. I think that the process started with an original arthropod, an original bacterium that came up with this toxin, and then as the arthropods diverged to form Coleoptera, Lepidoptera, etc., basically the bacteria tracked them. So, of course, this is a basic hypothesis, and yeah, it's going to take a long time to prove that.
Cool, very interesting. And I have a final question for you both. We thought that the genetic modified organisms with the BT toxin will be like a big salvation for crops. And now we have every time new reports of more and more and more insect resistant to cryotoxins. How do you see the future about that? Do you think there will be more organisms modified with different toxins, or we need to apply a different strategy?
Well, this is a difficult question.
Yeah, I know. I'm just asking for an opinion about that.
Yes, resistance. We will see more resistant insects in the field, especially because some growers in some countries, they are not following the rules. In order to avoid resistant development, there are some rules that have been followed, and the companies that sells the transgenic plants, they advised to use refugees areas without BT plants, so the susceptible larvae can grow in these refugees, and then the susceptible larvae, when they mate with the resistant larvae, the progeny will be heterozygous, and the heterozygous is susceptible. So if we have always refugee area where susceptible insects are there and can mate with the resistant insects, then the population of the resistant is lowered. I don't know if you follow my argument, but if the growers of the plants, the farmers, do not use refugees, then will not be susceptible insects around. So, and that's why the resistant goes very fast, because one resistant insect will mate only with one resistant insect, so the whole progeny will be resistant, and then it multiplies many times. Each female can have 300 babies, so that's why the resistant is become a big, big problem now in some countries, that they are not using refugees. Now in Mexico we have transgenic plants, we have cotton, but the growers use these refugees very strict, and we don't have any resistant insects in Mexico right now in the cotton area. But in the same plants, BT cotton, in India, they have very high levels of resistance, and in Brazil, very high levels of resistance. And the problem is that they didn't use these refugees. So if we introduce other characters like the beep throat proteins or other proteins, but the growers do the same, no? Without any carefully. So they have to take this technology with responsibility, and they have to use this with the responsibility, using these refugees areas to have the susceptible. They need to understand that we are dealing with a very complex problem, that is the resistant development, because the insects reproduce so fast and so abundantly that we cannot control them if we don't follow the rules.
To that, so we tried to scare you with all the resistance that's popping up, but not every crop and system with a BT plant has a resistance problem. And in fact, in the tobacco budworm, in the Heliothrus fluorescens, in the USA, there is still no resistance to the BT cotton, even the single gene cotton. And Bruce Tabashnik, whom Alejandra mentioned, has done a survey of cases of where there has not been a resistance and where there has been a resistance, showing basically a correlation. If a good resistance management strategy was followed, assuming following all of the recommendations, then you don't have resistance. The problem is when either farmers do not or cannot follow all of the recommendations. So you're trying to control evolution. You're selecting for resistance, but then you don't want to select too much. You want to select as little as possible and still get pest control. The strategy in the developed countries like Australia and the US, this works. In India, where you have many hundreds of thousands of poor farmers, each individually trying to grow their own crop, they cannot adopt this strategy. So it's inevitable that they would get resistance to the single gene cotton. And so the cotton with only one cry protein in it. So we are going to see resistance anyway, but now the companies can say, well, if we find two other proteins that have a completely different mode of action, the Cry2A protein that targets a different a different ABC protein, and a third protein, the so-called VIP, vegetative insecticidal protein, that has a completely different mode of action. We put all three of those insecticidal proteins in the same crop, then the probability an insect could only become resistant if it had mutations in all three receptors at the same time. So that is the strategy that the industries are taking. This would work for the major crops where they will invest the effort to do that, and also the markers of where they can control the grower and make them use these crops. Most cotton production in the world is done by very smallholder farmers that will not be manipulated in this way. So we really do have a challenge. And technology is not the only solution to this challenge.
Great. Thank you very much to both of the speakers. And please, to the audience, stay with us for the next panel in chemical ecology and plant science. Our next moderator is Ana Erika Gutierrez Cabrera. And that's it. For you, we have a small present from CONACYT, so I will give it to you. Thank you very much.
Hi, good morning. Welcome to the second part of the session, Chemical Ecology and Plant Science. I'm Ana Gutierrez, and it's my job in this time to coordinate this session. In this session, we have two speakers, Dr. Wilhelm Bollen. Dr. Bollen received his PhD from the University of Munster and Cologne. In 1997, joined in Max Planck Society and became director of the Department on the Bioorganic Chemistry in the Max Planck Institute for Chemical Ecology in Jena. His major fields of research covers sequestration of plant-derived compounds by the leaf beetles and plant insect interaction, with a special emphasis on induced defenses in plants. Our second speaker is Dr. Gabriel Olmedo. Dr. Olmedo obtained a PhD in microbiology from the University of Pennsylvania. Since 1990, she has been research as Investab Unidad de Irapato in the Department of Genetic Engineering, and is currently director of this unit in that unit. Sorry. Dr. Olmedo directs research group on molecular biology and molecular microbiology ecology. The group's interest is understanding how microbial community are assembled, and their experimental approach range from classical microbiology to genomics. So for each speaker, we have 20 minutes, and then we will have the question and answer session. And thank you, and now is the time for the Dr. Wilhelm.
Can I have the first slide? Okay, ladies and gentlemen, dear colleagues, it's a real pleasure to be here and to talk about science, which is at the frontiers of the science, as the title of the symposium says. I'm doing this in the Max Planck Society, and I should really emphasize what David Heckel meant. It is a continuous financial support of the society, which is independent of political or societal influence, which enables us to do research, cutting-edge research, at topics which usually not interesting or not relevant. And I want today to talk about the microbiome of an insect. The insect is a generalist herbivore, Spodoptera litoralis. These insects have been already mentioned in the talks before. These insects here are severe plant pests. Spodoptera litoralis in particular, because this animal can feed on 40 different plant families. That can really make severe damages. And it's endemic in Northern Africa, in Middle East, in the Mediterranean area, and is also invasive in the United States. What is shown on this slide here seems to be very simple. An insect feeding on a plant. And that means this is probably a very simple process, but it could be much more complicated than the first glance says, because we have to consider there's bacterial communities on the plant surface, a microbiome of the plant surface. There's another microbiome on the insect surface, and the most important microbiome, of course, is the collection of microbial cells inside the gut of the insects. And it might be very interesting to understand how these microbiomes are regulated, how they accumulate, and how they are composed. And that's exactly what we try to do with the eyes of the chemists. We want to see what controls the microbiome, what kind of chemistry produced by the microbes, produced by the insects, is relevant, and we finally want to see, is there a molecular interaction between the insect host and the microbes? To give you some impression about the life cycle of Spodoptera litoralis, as usually, life begins with the eggs. Then there are a number of days, and then we have the different developmental stages of this insect up to 6 larval different instars and moltings before the insect comes to pupation, and then a couple of days, finally, the adult species appear. And what we see here, over all these stages here, the insect is feeding on plants, ingesting plants, and the question is, how does the microbiome of the insect contribute to the digestion of plant material? Is there any change over all these different molting changes which we see here? And that is how we started our work, that we first began to analyze what is the composition of the gut microbiome at the different stages. When we come to day 0, when the insects freshly hatch from the eggs, we have a composition with several hundreds of different bacteria, but it's only a handful of different microorganisms which is dominating in the insect gut, of the early insect gut. There are a few enterococci, there are a few Clostridia, and this is everything which is present in the first larval instar here. But what we see when we come from the first to the second larval instar, there's a real dramatic change in the composition of the microbes, and it's Enterococcus muntii in this case, which begins to dominate. And then we have the next larval instars, the third, the fourth, and the fifth, where increasing numbers of Clostridia begin to contribute to the gut composition. We see here that this is a very regular and very reproducible change of the environment and reproducible change of the, let me say, microbial composition in the gut, which is important that we work with artificial diets under very controlled conditions. And I will show that on the next slide why. First of all, we see the microbiome on the surface of the insect is entirely different. And if we go and change the artificial diet, which is shown with the microbial composition here, to real plant material, then we see that the Clostridia again begin to accumulate very, very strongly. And since here the digestion of cellulose material and so on is very important, it is apparently the change to the plant material which causes this additional change in the gut microbiome. And therefore it is very important that microbiome studies are done under controllable and reproducible conditions using an artificial diet. And we see here very clearly the influence of the plant can be not ignored. Individual compounds may play a decisive role. In the meantime, we have analyzed the microbiome composition over the whole developmental cycle of this insect. And again, we have some pictures here. In the eggs, we have only the proteobacteria dominating, gram-negative bacteria, and only a few firmicutes here. These are the ones which are relevant for the digestion. If it comes to the early larval stages, that is corresponding to that what you have seen before, the firmicutes, and that comprises the Clostridia and the Enterococci, begin to dominate, and in the large larval instars, in the last ones where we have very strongly dominating the firmicutes, and the same is the case in the pupa. And then, when the adults emerges, we have, again, a completely different composition of these microbiomes, but interestingly, we have a clear difference between male and female adults. What is the reason on the chemical basis, of course, is not known. If we analyze which bacteria are where in the gut located, we have here a schematic drawing of the gut of Spodoptera littoralis. We have the foregut, the midgut, and the hindgut. And if we see at the distribution we see here, that in the foregut there are a few Clostridia, the majority are different Enterococci. The midgut is populated almost exclusively from the Clostridia, where most of the digestive work, cellulose degradation and so on is done. And when it comes to the hindgut, again, Clostridia are reduced and the Enterococci begin to dominate. And here you see that the insect gut is really a hostile environment. Here is a preparation of a Spodoptera gut, and we see that in the foregut area, we usually have a pH around 10, up to 10.5. The whole foregut area is strongly alkaline. Even the midgut is strongly alkaline, only the hindgut finally ends up at neutral pH. And of course, the microbes have to adjust to all these different pH gradients within the insect. Now the question comes up, what are the factors, or do we know factors which control already these microbial compositions? And one of these factors which has been recently identified is this compound here, the 8-hydroxyquinoline 2-carboxylic acid. This is a tryptophan metabolite which we discovered in the insect gut at very, very high concentrations. On average, the concentrations of this compound are about 1.5 to 5 millimolar concentration. This is really a huge concentration. What is known about this compound is usually a non-toxic chemical, but it's a strong chelator of metals, for example, iron, cobalt, nickel, and so on. For many of them, X-ray structures are known. And the most important finding is that this compound here is synthesized by Spodoptera littoralis inside the gut. And as additional information, I should say, these materials are de novo produced in the foregut midgut, and they are excreted with the feces. So there's an enormous investment into the turnover of tryptophan as the original compound into this compound, and then it is apparently wasted. And that means this compound must have an important biological function to justify this investment of the insect. No? This type of hydroxyquinolinic acid is not absolutely unique. They have been identified in the 60s already in the defensive secretions of water beetles. They are characteristic for this insect here, which is important for the distribution of malaria. The xanthorrhenic acid is a 5-hydroxy derivative of this compound. Finally, we have another 3-hydroxy derivative, also known from microorganisms. And finally, we have a quinolobactin, another siderophore, in this case from Pseudomonas fluorescens. So the compounds are not unique, but they are important to control the gut microbiome, as I will show in a few seconds. Here I will demonstrate that the distribution of this compound is not all over the insects. If you look into the lepidopterans, the occurrence is reduced to the noctuids, the lepidopterans, and so on, particularly the Spodoptera species, Heliotis species, and so on. There we find these high concentrations of the compound. And now I come to the ion chelation, which is apparently the principle by which these compounds act. Here we look for the concentration of this compound over the life cycle in the gut of the insect. You see, it begins already from the very beginning, goes up to very high concentration in the 6th larval instar just before the pupation begins. In the pupa, there's almost no residual hydroxyquinolinic acid, and in the adults, the concentration is also very low. So over the whole life cycle, the insect is full with these compounds. And if we now study the chelation activity and the effect of the chelation on developing microbial consortia, we look here. Here we have the Mycobacterium arborescens, also isolated from the gut of Spodoptera. That is a normal growth curve here, but if we do it in the presence of hydroxyquinolinic acid, at the lowest concentration which we find in the insect gut, the growth is almost completely inhibited. We see the same for Bacillus thuringiensis, which we have already heard before, complete inhibition of the growth all over the growth period. And very interesting, if we do the same experiment with Enterococcus muntii, we see that in the presence of even 1.5 millimolar hydroxyquinolinic acid, there's only a weak inhibition of growth. And that represents exactly the situation which we have in the gut of the species. That the hydroxyquinolinic acid is only an ion chelator but not a toxin is demonstrated here. Here it is an example with Escherichia coli. This is in black is a normal growth curve. If we add hydroxyquinolinic acid, the growth of the bacteria is strongly inhibited, but if we add iron, then of course the growth can be completely restored. And that clearly shows that hydroxyquinolinic acid is not toxic. The principle of inhibition is indeed ion chelation. A few slides to the biosynthesis of compound. I mentioned that the compound is produced in the gut from tryptophan by oxidative degradation. I will not discuss the biosynthetic pathway here, but I will draw your attention just to this hydroxylation step here, the quinorenine monooxidase. Since this compound, the xanthorenic acid, which you have seen before, is important for the growth of Aedes aegypti with the malaria spreading, chemical industry has already developed inhibitors for this biosynthesis 20 years ago, and one of these inhibitors of this KMO enzyme is this nitroalanine, which can be easily synthesized. And if we add this compound to the artificial diet for the insects, we can see that after a dosage of about 500 micrograms or 1 milligram per day, after, for example, roughly 2 weeks, where we have a highest concentration of H2A, the reduction at 1 milligram per day is up to 80%. And if we do this 80% inhibition and then look to the diversity of the microbes in the gut, we see there is a low diversity in the controls where the high concentration of the H2A is there. And if we do the inhibition experiments, we see from the color code here that in all these cases, the number of different bacteria directly increases. And that means we have here a functional evidence that the control of the microbiome is done by the insects by synthesizing the ion-chelating H2A. And very recently in David Heckel's group, they succeeded to knock down this KMO enzyme by CRISPR-Cas, and we will be now able to reproduce this experiment once again without the often unknown influence of the inhibitor. And that brings me to the next topic. Here we have seen the influence of the insect, and now we will see the influence of the microbes. I go back to the developmental picture from the very beginning. We see in the first larval instar, we have at least 3 different types of enterococci. And if we go to the second larval instar, already Enterococcus monti is completely dominating the composition of the microbiome. And that gave us thinking, and we thought, what might be the reason that all the competing enterococci disappear? And that they disappear is very important for the insect, because Enterococcus cassilieflavus and also Enterococcus faecalis, both of them are entomopathogenic. What happens after an infection with Enterococcus faecalis of the Manduca sexta larvae, you see here, they simply die. And therefore, the next data which I show here are very important. What we did, we cultured the different enterococci and took the supernatant, for example, Enterococcus montii, and added that to a growing culture of Enterococcus cassilieflavus. And you see here the inhibition zone. The same is true if we add the extracts to Enterococcus faecalis. Again, there's a strong inhibition zone. So something is produced by Enterococcus montii which kills the competitors, but which has no effect, as you can see here, on the Clostridia. And again, this reflects the composition of the gut microbiome, which at the very end is composed out of Enterococcus montii and, of course, Clostridia. So what is the factor? We isolated the factor. Here's the characterization by HPLC-MS. The MS is shown here. It's a peptide with about roughly 4,300 Dalton. It's a cationic peptide, which is demonstrated here. It's a so-called bacteriocin, which we called finally munticin. All these bacteriocins are not uncommon. They are quite widespread. They are produced by several bacteria to compete with neighbor bacteria in the fight for living space. And exactly like this happens here, Enterococcus montii produces this compound called munticin, which is apparently able as a cationic peptide to assemble in the membrane of the bacteria to form pores, which we have already heard several times today. And through these pores, of course, the bacteria are finally killed. That is the mechanism which has been found here. So in summary, we have two factors identified, the HQA as an insect-derived factor which controls the early microbiome composition, and with the monticin, a peptide which is produced by one of the major contributors to the microbiome, which kills all the competitors. I will introduce into a final series of slides the current methodology which we developed to have a holistic approach to see what kind of chemistry is going on in the gut, what are the interactions between the microbes and the insect host. Once again, we see here the whole developmental cycle. And the idea was to take, for example, Enterococcus monti, which is permanently present in the insect microbiome, to make it fluorescent, GFP labeled, and then to incorporate it into microbiome and let it go through the whole environmental development of the species. And then the idea was it should be possible to recover this fluorescently labeled bacteria and finally to do transcriptomics to see what is the response of the microbe to the diverse conditions in the insect gut. The approach has been very successful. We have developed a GFP-labeled Enterococcus. You can see here that the different larval instars show the increasing number of the Enterococcus up to the 5th instar. Then something happens. The insect begins to secrete cationic peptides to kill all of the bacteria, to prepare the pupation, and even in the pupa we see a spot of GFP-labeled Enterococcus. Interestingly enough, we see them also in the others, and what we could demonstrate by microscopy very carefully, we see them also in the egg. And that is one of the ways how these are translated into the next generation. So how can we do transcriptomics? In a few slides, we have taken out the gut from the species, we collected them, and then we used here the cell sorter system to recover all the fluorescent bacteria, which is easily possible, shown here. Then the DNA is extracted, and finally we can construct really the transcript data, which I will not interpret in detail right now, because this is currently summarized, but it will show in the next slide here that in principle the whole sequence which we have developed recently is in fact working. We see stress responses with components which we have here. We see quorum sensing upregulated in the microbes and so on. This is a systematic approach to see what is the response of the bacterium at different positions inside of the microbes, inside of the insect. And at the same time, we have done transcriptomics from the gut membrane of the insects, and we now begin to compare these two different transcripts to understand whether there is a dialogue between the two types of organisms. And the last slide shows that we indeed can expect something like a dialogue. What we have done here is a cultivation of Clostridium cellulolyticum, which also occurs in the insect gut, probably responsible for the digestion of cellulose in the midgut of the insects. And what we see, when we cultivate Clostridium here and add gut material from the insect, then this leads immediately to the production of new proteins here. And more importantly, if we add hydroxyquinolinic acid, this component alone leads to the production of the same new proteins here, which we could identify as part of the so-called cellulosome, which gives the idea that the insect reprograms the Clostridia to enhance the, let me say, cellulose degradation during the life cycle. So what we have to see here is a lot of possible interactions which we will hopefully be able to interpret more by using these, let me say, fluorescent spies inside the insects. And with this, I'm at the end. I have to thank a few co-workers, in particular Liang Ping. He did all these evolutionary, developmental changes of the microbiome. Yongxi Shao, who developed the multisin discovery. Beng Sonte and Tilotama Zumba are those which are currently developing this spy technology that we incorporate fluorescent enterococci into the insect gut. And finally, I have to thank Christian Hertek and David Hecke for a wonderful cooperation over many, many years. Thank you for attention.
Thank you for the presentation, and now it's time to question session. Sorry. The first question. Okay, thank you. Dr. Holben, have you looked for microorganisms such as fungi or protozoans? Have you looked for microorganisms such as fungi or protozoans?
Yes, we have looked for fungi in the gut. Their number is quite limited. It's about 5% of the species we have in bacteria. But we did not analyze the fungi for function.
Okay, thanks. The other question, Dr. Wilhelm, how involved is the diet of Spodoptera and the symbiosis with other organisms for the synthesis of 8-HQA?
So the diet is independent or has no influence on the concentration of the HQA. The diet is very important, as you have seen in the first slides, that as soon as we move from the artificial diet to plants, the composition of the gut microbiome changes. What we have seen over the years is that individual secondary metabolites of the plants can kill individual bacteria of the microbiome. So therefore, the constant conditions to analyze microbiomes are very important, and as only tryptophan is necessary as precursor for the biosynthesis of HQA, this is independent of the diet.
Okay.
The diet normally has no enhanced amounts of tryptophan.
Okay. Other question for you. When you do single-cell transcriptomics, how do you make sure that the transcriptional change are not caused by the process on the strand of slided cells?
Can you repeat it once more?
Okay. When you do single-cell transcriptomics, how do you make sure that the transcriptional change are not caused by the process of the stress?
Yeah, one has to be very careful with the processing. First of all, we have to open the insect and then to harvest the bacteria very fast and at cold temperature, that means around 0 degrees. We want to avoid that the transcriptional changes change again over the period. Then, after that, the microbes are dead and are sorted out and no further change comes into the game. The control is very important in this aspect, but it's difficult here because there are so many individual steps which also involve amplification of the DNA and so on. So one has to be very careful. We have done no quantitative changes up to now. That's something which is going on currently.
Okay. For Dr. Almedo, have you explored the possibility that the bacteria share plasmid that allow them to resist to antibiotics?
We have looked for plasmids, and 25% of our strains have plasmids, but we have no idea if they are directly involved in this thing. Yeah.
Okay. And what is the protection status for Cuatro Ciénegas?
Well, it's a conserved area, but the problem is that there is all this permission to use water, and these permits, are 100 years old, and are very difficult to change. And water is used for agriculture, so it's also sort of a debate, whether you can ask the people that live on agriculture there if they should turn off the water. The problem is that the way that water is used is not in the most efficient way. So it's really just, the valley is just dying away. And yeah, not much, we can do. We thought that the science was going to convince politics of the importance of this particular environment, but it hasn't happened so far.
Okay. Dr. Boland, do you find other or same pathogens against Spodoptera?
What? Once again, please.
Do you find some or other pathogens against Spodoptera?
Yeah, of course. These are not the 4 or 5 bacteria which I have discussed here. These are the 4 or 5 most dominant bacteria in the microbiome. Overall, there are several hundred of bacteria, a lot of pathogenic bacteria. I have discussed here only the entomopathogenic bacteria from the genus of Enterococcus, but there are many, many more.
Okay. The last question is for Dr. Boland. Could a bacterium enable an insect to feed on a toxic plant?
I did not understand. Once more.
Could a bacterium enable an insect to feed on a toxic plant?
I didn't understand.
I think that can an insect, feed on a plant that is toxic, because the bacteria is going to probably give it protection.
Okay. This is a topic which has not been studied as yet, but it is of course clear. In the moment where the insect is feeding on the plant, it inserts by feeding bacteria inside the plant. Then the plant has, of course, a lot of defense mechanisms, salicylate and so on, pathways, which are there to kill the bacteria and to prevent entrance of the bacteria into the plant. Okay. But of course, some of the bacteria will survive, some of the bacteria will upregulate plant defenses.
Okay, thank you. Thank you, Bar. If there are no more questions, we thank to the speakers, and thank you for this session. And the CONACYT give you now it's time to a break for 15 minutes, and we return here with the next session.
I'm the moderator of this session, Computational Sciences. Thank you for being here. Now it's my honor to present Dr. Humberto Salazar Ibargüen. Professor Humberto Salazar is a researcher in the field of particle and extraparticle physics at the Benemérita Universidad Autónoma de Puebla in Mexico, where he pioneered more than 20 years ago. The participation of Mexico in large international collaborations, such as the Peer Auger Observatory, the compact muon solenoid located at the CERN laboratory in Europe, among others. He is also a member of the National System of Researchers in Mexico with the highest level and the Mexican Academy of Sciences. And today, he will sharing with us Mexican Supercomputing Network. And another special guest, Stefan Hensel. Stefan Hensel is director of the Max Planck Institute for Computing and Data Facility and is a member of the Council for the Computer Infrastructure of the Max Planck Society. He studied computer science at the Technological University in Munich, earning a diploma in informatics. His research interests include operating systems, scientific computing, parallel and high-performance computing, and file systems, among others. The topic is towards extra computing. For health reasons, he cannot be here with us, but Dr. Marcus Ramp will be on his behalf. Please, Dr. Humberto. Thank you.
Good afternoon. Thanks a lot to the organizers for inviting me to present the status of the National Supercomputer Center at Puebla, LNS for short. As you have heard in the presentation, I work in the high energy physics area, but we are users of supercomputing. We depend a lot on supercomputers, and I am in charge of this national laboratory, which I will present in this symposium. Before details of this laboratory, I want to mention, because I should mention the status of the supercomputing in Mexico, the network of supercomputers in Mexico. I will say that this laboratory is not the only one in Mexico. In Mexico, there are at least 5 centers of supercomputers. This is one of them. The other 4 are one in San Luis Potosí, the National Center of Supercomputing, the Delta Metropolitana, which includes the facilities at the UNAM, the CIMBESTAB, and the Metropolitan University, and also the Abacus. This is the Mexico State facility for supercomputing, and in very short time we will have also the supercomputer at the University of Guadalajara, which will be the largest one at Mexico. I can say that all these facilities, this network of facilities together can reach a performance at least of 1.5 petabytes, maybe 2 petabytes, and this is they are the network of facilities in Mexico. It's not too much, it's not too much like in Germany, but we are working in the center, and I think, as I will present in this talk, we are ready to have the petascale facilities and maybe to collaborate in the exascale experience. Okay, I will present first of all the LNS infrastructure, I mean the support infrastructure for the computer, then the details of the computer and the applications in high energy physics that we are, what we are doing in high energy physics. The infrastructure of our laboratory is certified by the Safety World-Class Quality Assurance Data, has Safety World-Class Quality Assurance Data Center, by the International Computer Room Expert Association, ICREA. The level of certification is 3. In the university is the higher level that we can find in Mexico. Its capacity is of 10 racks, each one with 42 units, and the storage capacity is 1.5 petabytes. The certification, we reached the certification in May of 2016. The support infrastructure, that means the electrical room and the support for the computer, is a redundant system N+1. We have an electrical room, we have the cooling system, and we have the UPS of 160 kilovatios each one. As I said, we have every system redundant, and that includes also the electrical line. We have 2 supporting electrical lines. Also, we are working on the communication network in order to have a optical link with other laboratories, like a large millimeter telescope, and also the high altitude water Cherenkov detector, both of them in the Orizaba Peak, 60 miles far from our facilities. This fiber optic connection is in progress, and we have ready this connection in this first semester of this year. Our external link is also redundant, and we have 5 + 5 gigabytes optical link. Regarding our main computer, this is the Xeon processor. We have Xeon processors. We have 268 computing nodes, with several of them with different RAM memory, in order to offer different service for different academic groups. And the internal connection of them with the storage part is very high velocity. We have InfiniBand interface, a switch with which can, the velocity is 56 gigabytes per second. We have also 6 special nodes, 2 of them with NVIDIA GPUs, 2 multiple integrated cores, and 2 fat nodes, each one with 1 terabyte memory RAM. All of them are connected with the storage of 1 petabyte. This computer can sustain a performance in link packed text until 200 teraflops. And also, because the modern technology, this is in the green 500 group of computers. The second computer that we have is with technology of many integrated cores, mix, and is of the type of Xeon 5 Night Landing processor. This is a new technology. We bought this computer 2 years ago, And in only one rack, we can reach and performance of 135 teraflops. This is the peak performance, and as I mentioned, is in only one rack. So you can compare the other computer that we are using 6 racks for 200 teraflops, and in this one we are using just 1 rack. In this new technology, we can run highly parallel applications, not only for academical purpose, but also for the industry and business. The internal connection is faster, is 100 gigabytes per second. The storage is almost 1/2 petabyte, and the number of cores is about 3,000. The more recent mini cluster that we have bought last year is GPU's processor, and use very cheap GPUs, CUDA cores, and reach also a very good performance, 90 teraflops peaks, and this used for very highly parallel activities. So we can summarize that the infrastructure that we have in computer at the National Supercomputing Laboratory are 3 computers, one that can reach 200 teraflops, another one that can reach 135 teraflops, and the new one, GPU's cluster, 90 teraflops. If we have highly parallel applications, then we can use the 3 of them, and we can reach more than 400 teraflops performance. This is the infrastructure that we have, and we are giving support to academical groups, scientific and technological research, not only of the original consortium, that is the Benemérita Universidad Autónoma de Puebla, the National Institute of Astrophysics, Optics and Electronica, and the University of the Americas, and recently the University of Chiapas, but all the national institutions. And also we have a dedicated office that which have a business plan and have a service portfolio in order to offer paid services. These are the logos of the academical institutions that are using our laboratory. There is more than 25 institutions working with us in academical research. And the application software that we can run and we have is not only open source, but also paid software like Gaussian. This is the most popular software for our users, Gaussian, MolPro, TeraChem, also that is for chemistry, for material science, VASP, Crystal, COMSOL, Lumerical, for biology and physiology, Civil, for high energy physics, Topaz, and also mainly open source softwares. And in other areas, we have Mathematica, MATLAB, Intel Parallel Studio, PGI, CDK. Our users, we can define several areas like nanotechnology, which is using 17%, material engineering, which is using 15%, biophysics, which is using 11%, and high energy physics, 10%, chemistry, 10%, semiconductors material, 10%. These are the uses of our laboratory, and I will give the details of how people from high energy physics is using this laboratory. Then, first of all, the main users of our laboratory are large collaboration, international large collaborations, like the collaboration that work with the group of compact muon selenoid, the CMS, the Mexican group, or at least part of the Mexican group, use our laboratory to make simulation and data analysis from this experiment, not only in the main computer, but also in the new one based on GPUs. For example, you can see here the modeling of decay of heavy Higgs boson, decaying into light bosons. This is one of the graphs that takes normally 16 days in normal computers, and in the new cluster, mini clusters, take just 20 minutes. In this experiment, we have at least 121 published papers that mention that LNS is a support supercomputing center. Another important experiment at the CERN facilities that use the LNS is the ALICE project. This project is very important in Mexico. The Mexican collaboration make a lot of things in this experiment, and particularly work in the study of high muons multiplicity cosmic rays event, and also in the trigger, in the performance of fast interaction trigger for the ALICE upgrade. Recently, they discovered that not only in the very heavy ions like lead, it's possible to have a fast transition, but also in the proton-proton collisions when we have a high multiplicity of these events. In that case, we can have also this transition to a phase in which quarks and gluons are not confined. This research was published in Nature Physics last year. Okay, the other large collaborations that are user of this laboratory is the Cosmic Ray community in Mexico, and the main observatory which are making data analysis and simulations are the Pierre Observatory and the High Altitude Water Cherenkov detector. The last one is at the Orizaba Peak, and will be connected direct by fiber optics with our laboratory. I will talk then about the extensive eye showers and the Pierre Observatory, and how to use in the modeling and the simulation of these showers, how to use the simulation of these showers needs a lot of computing time if the energy increase. Then we have a complete model and system to have knowledge about these showers, and to make data analysis with our observatories. Then I will skip some of this. But one important thing is even having high-performance computing, if the energy of cosmic rays is too high, then it's not possible to run these simulations in these computers. So we need to make some kind of a compromise and to make a reduction of the statistics and to follow just some of the particles, some representative particles in the secondary particles. We use for simulate the detectors Jan4. This is free software mainly. And with these components, we can model the Pierre-Auger Observatory and the Hawk Observatory. And with simulation, then we can understand what is the energy, the mass composition, and the arriving direction of these cosmic rays. With, let me skip this, analyzing the data of these showers, and especially we can have a control room, remote control room. This is the control room at the University of Puebla. We can have in real time the observations of the observatory, and we can make simulations and then understand the main characteristics of these cosmic rays. With all these components, recently, Pierre-Auger Observatory also concludes that the highest cosmic rays should come from the should be from extragalactic origin. This is a science paper. Related to the Hawk Observatory, also we can simulate all the process of the photons coming to this observatory, the extensive air shower, and we study mainly photons, and also photons from very high energy coming for the very impressive phenomena at the university. So we simulate the observatory, we simulate the showers in the atmosphere, we simulate the response, and we can make comparisons of what is arriving to the observatory with these simulations. All of this process can be highly parallel, and because of that we can use our 3 computers in order to make simulation and data analysis. Okay, as conclusion, I can say that this national laboratory is a consolidated supercomputing laboratory and has become an important computing tool to do research in several scientific areas. The LNS plays an important role in high-energy physics. I have showed for large collaboration. And the data center and the network infrastructure at the LNS allow the optimal use of the computers. It's a certified data center and have a service portfolio for industry and business users. Thank you.
So, buenos dias. My name is Markus Ramp. I'm from the Max Planck Computing and Data Facility in Germany. I'm group leader for high-performance computing applications there, and it's a great pleasure for me to step in for Stefan Heinzel, who couldn't make it, unfortunately. I have to say I'm very impressed by the scientific diversity of the last 3 days, and I really enjoyed that. In particular, I enjoyed talks in fields which are quite far from my own kind of background, which is computational astrophysics. And in return, I thought we should make the talk as introductory and as general as possible, given that it is a bit technical by nature. And thanks for bringing my presentation up. So the topic is about towards exascale computing. So this is a frontier that the high-performance computing community is about to cross within the next couple of years. And I'll try to point out a few of the basic obstacles that are still ahead, ways to overcome them, and also point out challenges and opportunities for applications for computational sciences in particular. I'll say a few words about our own center towards the end of my talk. Now, just as a very gentle introduction, the basic metric to measure the speed of a computer is floating point operations per second, often abbreviated as flops. Quite natural, since this is by far the most frequent type of computation that is done if you do numerical simulations, and that's what these machines are being built for. It started during the Second World War, for reasons that probably most of you know, with something like 100 floating point operations per second, barely outpacing a bunch of humans. And then over the years, I should show this machine. So this is a historic picture of this ENIAC machine built by John von Neumann and collaborators. And then over the years, reaching a tremendous growth of kiloflops, megaflops in the 60s, gigaflops in the 80s were reached with a bunch of these iconic machines listed there, until the petaflops level was reached 2010, 10 to the 15 floating point operations per second. And here's an image of the machine which was operated in Oak Ridge, Tennessee. And we are close, as we think, to reaching the next kind of level, which is exaflops, 10 to the 18 floating point operations per second. Probably during the first half of the next decade. It's not yet clear which kind of machine it will be, and of course also not the name. Now, again, a little bit more quantitative. The ranking, and this was already alluded to in the previous talk, there's a famous ranking of supercomputers, the so-called top 500 list, which is just listing the 500 fastest supercomputers. And this goes back over time until the early 1990s. And what you can see here in these lines, this is a logarithmic plot. You see the fastest supercomputer here increasing at an exponential rate. The blue symbols here is the last computer in this list, number 500, and the green one is the sum of all of them. And what you can see here, and I think this is really remarkable when you see this. So, for example, if you invest a bunch of millions of dollars, many of, in fact, to enter here as the top one supercomputer in the list, it takes you something like 6 to 8 years to drop completely out of this, so you will not be listed anymore. And if you compare, for example, your smartphone would have made it into the list at least in the early 90s. What you typically have in a laptop or in a PC was number one supercomputer in the 90s. And you have something like a graphics card for a couple of hundred of euros would still be in the list until the early 2000s. Now, this is exponential growth over more than 2 decades. As a side remark, this is based on a very specific benchmark. And keep in mind that scientific simulation codes typically today achieve only a few % of this value. Now, this is a nice little story also to appreciate the enormous growth that has happened. So one of the first non-military applications of these early machines was numerical weather forecasting. And they were barely able to do a relatively coarse model, which you can see here, of Northern America. And it required 24 hours to do a 24 hours forecast, so at the edge. A few people took the effort to reconstruct the same kind of model, numerical model, re-implemented this as a Java application, run it on a phone, the same forecast, same resolution, and were able to complete this in less than a second in 2008. So, what is driving this? You may have heard about this, and it was even alluded to earlier in a talk about genomics by Johannes Krause, I guess it was. And this is an observation made by Gordon Moore in the 60s, observing that the number of transistors on a chip doubles roughly every 18 months at constant cost. And this has sustained from 60 devices on a chip, 1965, meanwhile to more than 20 billions of devices of a chip, which was introduced last year. And what you can see here is, again, over time here on a logarithmic scale, this exponential growth of the number of transistors. Basically, this was, from the technological point of view, this was sustained by just making devices or structures on a chip smaller and smaller. And we are currently at 40 nanometers. At 2005, something happened. So this growth here and the miniaturization was also going along with an increase in the clock frequency of the chip, which you can see here in green, which means that processors just got faster over time. This leveled off around 2005. So while the number of transistors was still growing, it was no longer possible to increase the clock frequency. And the response of the industry was to introduce multi-core processors, which are now everywhere in phones, in laptops, in servers. Which also meant that what was called a free lunch, you just wait for next generation of a computer and this makes your software faster, was over. So we had to go parallel since 2005. And if you look very closely, you cannot see it in this plot, this kind of increase, exponential increase, seems now also coming to an end. So there seems to be a kind of a flip here from an 18-month cadence to something like 24 months. So in order to do computations, you also need to fetch data from memory and write them back. And unfortunately, the memory performance has not kept pace with the computing performance over the years, which is illustrated here. Sorry. Which is illustrated here. So there's a widening gap between CPU and memory performance. And if you take the ratio of the memory performance and the computing performance, you get this number here, the number of bytes you can fetch from memory for each floating point operation. And we are currently at a level of 0.1, and this is expected to decrease according to this trend until we reach exascale by another order of magnitude. Now, for the experts already, this is a very low number. You would like to have it rather ideally at a value of 8 or something. And in fact, most of our algorithms and codes which are running on these high-performance computers are actually not limited by the capability of doing floating point operations, but by the performance of the memory. Unfortunately, improving this ratio, improving the memory bandwidth to computation ratio turns out to be very costly. There have been new technologies, they're introduced on the market, high bandwidth memory in the most general sense, but they're extremely costly and cannot completely replace the memory as we are using it these days. Now, just briefly, and this is often overlooked, the memory also contributes quite substantially and continues to increase to contribute to the total power budget of the chip. While this was something like a quarter or a third around 2012, this is expected to be roughly half of the entire power budget of the chip within the next couple of years. And a possible solution to overcome this is replace the traditional memory, which is built into computers these days, DRAM, by a combination of even slower memory, something which you know in principle from flash devices, from USB sticks. It's very energy efficient, can be mounted in huge amounts in principle, so it can be used for capacity. And in addition to this, equip the machines with fast, high bandwidth memory, and this is actually already being done in graphics processing units. But as you can see for the numbers, this is a relatively scarce resource. Now, talking about power and energy, if you look at the total power consumption, electrical power consumption of a high-performance computer, just to give you a first idea of the scales we are talking about, this is, again, over the years. Let's look at this or focus at this line here. This is the average of the top 10. So this is the real supercomputers in the list. And the scale here is megawatts, right? So this is not something small or negligible which you operate in a small hut or something like this. And if you would naively just take the machines which are currently being operated in our centers, which roughly, just to give you orders of magnitude, are at that scale, let's say 1 petaflop requires electricity power of 1 megawatt. And if you multiply this by a year, at least in Europe, with electricity costs, this is at least €1 million or even more. So even $2 million, maybe like something like this. And I think it's obvious that you cannot simply just scale this up by a factor of 1,000 in order to reach exaflops, because then, I mean, would you like to operate at a gigawatt? This is not the wind turbine scale, but just to give you the idea here, this is nuclear power plant scale, right? So what is needed clearly is that the processor, the intrinsic efficiency in terms of how many computations per watt can be done, needs still at least a factor of 20 to 50 in improvement in order to reach exascale, given that something like 20 to 50 megawatts is considered tolerable for powering such a computer. At least this seems to be the convention or the consensus in the US. I'm not sure about Europe in this respect. But still, I mean, this is more than an order of magnitude still to go. And there's hope. If you, again, look at one of these pseudo-laws, observations, more or less, what has been happening in the past, there's, in fact, an increase also in this metric in computations of flops per watt or actually it's watt hour here. Over the years, you see also kind of an exponential increase here and a little bit more, if this works, a little bit more quantitatively here and also noting a few specific processors or processor generations here. Again, over time, the efficiency in terms of gigaflops per watt And I marked this line here, which is 1 gigaflop per watt. This is, let's say, current situation, which I labeled with the wind turbine here. This would mean 1 exaflop at a gigawatt, which is clearly, I mean, not the thing we would like to do. 1 order of magnitude here is 1 exaflop. With 100 megawatts, still quite a lot, but this is something which current processors, not the regular ones, but the ones which are GPUs or similar to GPUs, like the Xeon Phi, which unfortunately is dead already, are able to reach already. And I think it is more or less clear also from this picture and other considerations that most probably an exascale machine will not be built with standard processors, let's say the Intel processors, which are standard multi-core processors, which are marked in blue here. But this will most likely be something like many, more or less lightweight, energy efficient cores, which gain their efficiency by having reduced instruction sets and also reduced clock frequencies. And what we essentially do here is trade complexity of these individual cores or processing units for their massive number. Unfortunately, this also comes at a price for the scientific programmer, and I'll come to that in a minute. And based on these kind of projections and considerations, we expect a system, a usable system, where you can do real science with something like 500 petaflops, half of an exaflop, with 60 megawatt in 2020, and there are plans and roadmaps in the US probably which seem reasonable there. And this is just a very rough sketch, more or less indicating that I mean the kind of trade space and constraints for designing such a system is very complex, and you might end up in a niche of a parameter space in terms of the available memory and memory speeds and number of nodes, which is probably not the most cozy one from the application programmer's point of view, unfortunately. A little bit more specific, and I'd like to focus you only on the colored kind of numbers here. How would such a system likely look like? Look at this. It will likely be something like between 1,000 order of magnitude, a few thousands probably, to tens of thousands of nodes. Thousands to tens of thousands cores per node, times, well, depending on the technical solution that is going to be deployed, and this is not yet clear or not yet even visible, times 1 million of these nodes, or just 100,000 of nodes if the individual nodes are more powerful. And what is also interesting to note here is that there's a high danger of failures, while current systems at the petaflop level are able to sustain computations or jobs for a day at least. You have a high chance of a failure happening during your job on such new machines. And for the applications of for scientific programming, the consequences are dramatic, as you can imagine. So it is far from trivial being able to handle this extreme level of concurrency, which we expect at least at the level of a few millions. Fast memory is a scarce resource, as I've pointed out. Managing data locality as a consequence is key, but is not yet well supported by programming models. We need fault-tolerant algorithms and applications, and also abstractions and modern programming techniques in order to remain portable and also flexible for strategic reasons. And the underlying assumption here, and this is important to know, I mean, major application codes such as those which are also being developed in the Max Planck Society, they build on many person decades of effort and in-depth expertise, right? So you cannot simply rewrite them. There's also challenges on the system software level, which I'm not going to go into much detail here. And well, you might ask, why do you need such big machines? I mean, they're expensive, they're difficult to use. I probably don't have time to go into much detail here, but basically this is the quest for more physical, chemical, biological, whatever realism. And there are basically two kind of areas. This is numerical accuracy. Most of the algorithms which are around have a reasonably bad scaling with the size of grids or with the resolution or the number of particles, so that it can easily fill bigger machines and would like to fill bigger machines. The same is true for relaxing model assumptions, mainly driven by the need or by the motivation to go to first principles and relax assumptions, simplifying assumptions, which might be or are, in fact, in many of the simulations which are being done today. And in fact, to be a little bit more practical and give you the view what is really being done, we asked around in Max Planck Society about the vision, what people would like to do if they had access to machines like that. And just to give you a few examples, Professor Grubmüller from biophysical chemistry, for example, would like to simulate the millisecond in the life of a virus, proteins, other things. Volker Springel, Max Planck for Astrophysics, would like to simulate the Milky Way and at unprecedented resolution, for example. And, for example, also would like to and thinks this is also feasible, perform more reliable calculations of, for example, a problem like cosmic ionization. Similar in climate sciences and plasma physics, grand challenges, and they expect really breakthroughs in their fields. And of course, there are many more: numerical weather prediction, combustion, materials science, data analytics, and many more. Just a very brief kind of summary here. So exaflop systems are at the level of a few $100 million in cost, are on the horizon, and we expect the technology to trickle down. Petaflop, petascale, order of $10 million is mainstream, more or less, and you can do teraflop simulations on your desktop these days. This implies, I mean, the challenges I had pointed out implies that new algorithms need to be developed. You need long-term interdisciplinary efforts by teams of computer scientists, applied mathematicians, and computational scientists. And this is actually already being done in the Max Planck Society. And even if you're not interested in the very largest machines, keep in mind current supercomputers will be the future mid-range clusters, and I couldn't resist doing this comparison here. So this is again the Jaguar machine filling a room which is at least this size here. I saw it myself 2010. Max Planck has a bunch of boring racks, more or less, as you can see here, which basically is at the same level installed 2016. So from my point of view, this is very impressive here. If you allow me maybe 2 more minutes, I will just very briefly illustrate the role the MPCDF has within the Max Planck Society. So we are located south of Germany, very close to Munich at the research campus. We have lots of collaborations and hosting of clusters on top of a high-performance computing systems, and this is the HPC labels here, with Max Planck Institutes labeled here in yellow, and we are also hosting lots of data and data repositories for the Max Planck Society on top of a high-performance computing system, and Max Planck Society has not its top priority to show off in the top 500 list, which is very good, I think. But still, I think it's nice to see that Max Planck Society has been represented with only one single exception here over the years in the list, and we are about to actually next week power up a system which is at the level of 10 petaflops and will likely enter the list among the top 20 maybe in the next issue. The group I'm responsible for collaborates with the domain scientists in the institutes in the Max Planck Society to develop and optimize codes. I think this is one of the strongholds also of Max Planck Society, that they have a number of renowned high-level application codes in which are leading in their fields. In addition to the high-performance computing activities, data has grown a lot. This is a ranking of the data storage capacity of senders. I'm not asking you to read this, I just took a few numbers. So there's a European Center of Medium Weather Forecast with 36 petabytes. Brookhaven and MPCDF is already here, I think at number 6 or 7 with 78 petabytes. Well, and I should point out that the growth is these days mainly, and this was probably also obvious from the talks or from a couple of talks during the last 3 days. The growth recently mainly comes from the life sciences, biology, medicine section, specifically in the Max Planck Society. And of course, you might say storing data on tapes and counting the bytes is probably not the most exciting thing from the scientific point of view. But this tells, I think, that Max Planck Society and their partners are among the top data producers worldwide, and analyzing these data and making them available to others, sharing, annotating this, I think is a huge challenge and another frontier of science which needs to be tackled during the next couple of years, and we're looking forward to this. So I thank you for your patience, and thanks to everybody who stayed until the very last talk. Thank you very much.
Now is a session of questions. So please, Dr. Ramp, what's your opinion about quantum computers? Oh, this question is mine too, because I'm working in quantum computation. Please.
This is a very hard one. So I think there's still some years to go until it comes to a level where you can do reasonably general purpose computing. So I do not expect that, let's say, for doing numerical simulations, this will kind of enter our field within the next couple of years. Still, I think it's a very exciting development, and there's also activity, scientific activity in the Max Planck Society to look into this. So this is closely observed and maybe one of the next revolutions about to come.
Okay, thank you very much. In your institute, is there any investigation about it? Yes, in Max Planck? Yeah, about quantum computers.
We are not, I mean, not specifically in our center. I'm aware of first collaborations. So, for example, Professor Scheffler at the Fritz Haber Institute has just launched a collaboration with IBM Rüschlikon to look at quantum computers. I know that we have at Max Planck for quantum optics. There's Ignacio Siriac, who is a leader in this field, and as far as I know, they are just forming a group of people who is pushing this within the society. But probably I'm not the best to ask about this question, because this is really very, very new and well, not fully developed activities.
Okay, and another question in relation with quantum computing, I think is a very interesting topic. This question is for Dr. Salazar or Dr. Demi. Does quantum computing have a future? Please.
As we say, this is in the very early stage of evolution. At least, for example, for us in Puebla, we have just one researcher working on that topic. So I think have a lot of future, but not in the near future. I think we should wait at least 10 years more to have some results and some progress in this area. That is what I think.
I have a very short answer, yes.
Okay, thank you very much. And here is a question for Dr. Humberto. Are all the systems opened for academic research groups? I think related to the quantum, the center you have.
Yes, this is a national laboratory, and we make 2 calls for proposal every year, and this is complete open. We have a ad hoc committee to evaluate the proposal, and depending on the evaluation, we can give some time of computing time.
Okay, there's another question for Dr. Marcus. What does it mean the free lunching is over? Can you talk more about it, please?
Yes, I mean, this was the title of a paper. I don't think there was ever a free lunch, but before this 2005 kind of turn, we had a single processor, right, which was growing faster at an exponential rate over years. So what you could do, and I still had the chance to have a few years in this era, it was much simpler, right? You had your software, and when a new generation, when Max Planck had its new generation of supercomputer, it was still just a few processors, and they were just faster, right? Nowadays, you get just more and more and more cores or processing units, so you have to work hard to parallelize your codes, which is highly non-trivial, in order to take advantage of this speed increase.
Okay, thank you. Another question for you, please. Is the trend to go to free software or to go to commercial software? I don't know, free software or commercial software? The trend?
The trend, I think there is a trend, and I think this, from my personal opinion, this is a very good trend. There's a trend clearly in Max Planck Society and probably also beyond to make scientific codes available as open source and shared with others. There's a cost to this, and there's a risk to this, because, I mean, you open up, right? You have to support. You maybe lose some of your competitive edge, but I think on the long term, this is a very, very valuable kind of decision, not only for the society, but also for the code itself. It helps the quality, and it helps science. So I think this is a very good trend, which we do observe and we do appreciate.
Okay, thank you. Another question, I think maybe is for Dr. Salazar. Does extra telecommunication represent a barrier on supercomputer development? Does actual telecommunication represent a barrier on supercomputer development? I think the question is the actual telecommunication is limited. So if it represents a barrier on supercomputer development.
Okay. I think if the processes are running in the supercomputer, you need just a not so good communication to work in this computer. But if you need to transfer data very often, then it could be something like can stop the use of the computer. But I think in our case, I mentioned that we have a connection of 10 gigabytes. I think it's enough for a lot of data analysis and for a lot of projects. I think it's enough in this moment. But maybe for very special projects, it could be a problem.
Okay, here is another question, but I can't understand the letter.
I think the question is, and I think it's a very relevant one, how can misuse of public computing facilities for cryptocurrency uses be avoided? Do you have a solution?
No. No, we don't have solution, but we have talked about that in our group, how to prevent this kind of use of the supercomputer, because we have not enough tools to see what people is doing with our supercomputer.
Yeah, very similar with us. We are talking about this. It's easy to talk, but it's difficult to implement a reliable solution, and we don't have one, I have to admit. Maybe we are a little bit in a different situation because we are not a public computing center, so this is open only to Max Planck Society, and there's kind of a ring of trust in some sense, which is a weak argument in that respect, I admit. So one should be able probably to detect such abuse. But you should also keep in mind, I mean, there are also possible abuses, right? So tracing everything which is not real science is a huge challenge in its own respect, I guess. But we're trying.
Okay. Thank you very much. Please don't move. We will continue with the closing ceremony. Thank you. Oh, sorry. We have a small gift for you.
Hello, everybody. Thanks you for joining us until the end of this event. We really hope you are enjoying it. Now, it's my honor to present Dr. Andrés Repte and Dr. Julia Tagüeña for the closing remarks. Dr. Julia Tagüeña is Deputy Director General for Scientific Development at the Mexican National Council of Science and Technology. She has been a researcher at the Energy Research Center of the National Autonomous University of Mexico, UNAM, of which she also served as director. She studied physics at UNAM and obtained a PhD at Oxford University. Her main fields of research are solid-state physics, renewable energies, and science communication. She's a member of the Mexico's National Research System with the highest rank, and of different societies such as the Mexican Academy of Sciences and the Institute of Physics of the United Kingdom. Dr. Andrés Trepte is head of the Max Planck Society Liaison Office for Latin America. He attended to the Latin American Studies at the University of Rostock, Germany, and got his PhD in economics. Dr. Trepte served in different positions and institutions of the German scientific system, such as the German Council of Science, and since 1991 works for the Max Planck Society. He has a long experience establishing, maintaining, and evaluating research groups and institutions. He has also been involved in science marketing and communication, consulting in prospective studies on the development of science and building new international scientific cooperation instruments. He has been member in different committees of experts, such as the Federal Ministry of Education and Research, the European Commission, the European Science Foundation, the G7, OECD, among others. So now I give the word to Dr. Julia Tagüeña.
Well, it is a big pleasure to be here with you now for these closing remarks. We talk about this ceremony, Andrés and I, and we decided that we wanted to give you some insight of how we are already collaborating, some successes we have had in our collaboration with Germany, and also to show you some possible ways to go on in this collaboration. We have heard here some projects that are already being worked together by Mexico and Germany. Of course, we hope that many others will come from this meeting. I promise my friend Andrés Sandoval from the High Energy Water Cherenkov experiment that I will mention that not all the German-Mexican collaborations have been presented in this meeting because HOG has a very strong collaboration with Germany, but the German group couldn't come, so it wasn't presented here. So we have seen some of the good collaborations, but not all of them. There are still some others working that we are very proud that this is working so well between our countries. So we have prepared a very small presentation to show you some of these results between the Mexico and German collaboration. So if I can see the next one, please. Oh, I have here this. I'm sorry. I thought I had to ask for it. Okay, so let me talk about the joint postdoctoral program, which is precisely with Max Planck Society. And it just started in 2017. You can see there how the agreement was signed in April 2016, and it will go on till 2021. It involved, at the moment, we have invested 132,500 euros, and we are supporting 5 postdoctoral fellowships. The fields of knowledge that we are addressing are astronomy, chemistry, physics and engineering, biology and medicine, humanities, social science, and knowledge science, which I could have said everything, because really we are covering all possible interests. And these are some of the institutes that we have already sent Mexican postdoctoral fellowships there. The call was launched on July 27. We are giving here just some of the information that was on the call. This is a moment to tell you that it's really important to follow up the CONACYT website because we are continuously presenting opportunities there. And so it's important to watch up for some of these. This one is already closed, of course, but it's good to be really following it. Something else that is important to mention is that we have a bulletin and we send information to everybody, so you can also send us your information so you will receive this data about the new calls that we will launch. So this happened in August 2017, and we received 68 proposals. We selected 5. As all the postdoctoral fellowships that CONACYT supports, it starts with 1 year and it could be extended to 2 years if the first year it has a positive evaluation, and if the project is really worth following up another year, another extra year. As I mentioned here, you have the possible, how much money Mexico put on it, and of course, Max Planck is putting practically the same, well, really the same amount, but in different ways, because Max Planck, they have to pay for the administrative and research fees, and they also provide additional support. So we are sure that these fellowships, they can really have a very good life while they are working. Well, they have to work very hard, that is for sure. So these are the names of the first Max Planck CONACYT fellowships. As you can see, they are covering different subjects. Oh my goodness, what did I do? Oops. Uh-oh, here it is. As you can see, we are covering different subjects: chemistry, physics, evolutionary biology, breeding research, and anthropology. Actually, we wanted to have like a wide scheme of subjects, and of course, we always check. I mean, we are choosing by excellency, but we always try to find that there is also some parity, some gender parity. So these are the 5 postdoctoral fellowships. They all are starting in 2018. On the other hand, as you know, CONACYT have, well, you should know it by now because all the moderators of the sessions belong to this program. Mexico has had a very successful program to give scholarships for training. Just a month ago, it was given like a special diploma to the 400,000 scholarship given by Mexico, by the people of Mexico, to get a PhD, to get a postdoctoral degree. I had a scholarship from CONACYT some years ago, of course, and now there are 400,000 Mexicans that have been supported by this program. So it was important to follow the program with a program to hire some of these people, because of course many of these postdoctoral students, they go to different universities, but it was important to have like a parallel program that is called Cátedras CONACYT, which is similar to the CNRS that CONACYT hires, but then commissions these researchers in different universities. We invited some of them to be moderators during this meeting, Max Planck CONACYT, and we have made an effort to use the program to hire Mexicans that have been doing a postdoctoral stay in Germany. At the moment, we have already 4 of these people. You met some of them already. So one, Arturo Iván Gómez, did his postdoctoral studies on radio astronomy, and now he's hired by INAOE. Asdrúbal Burgos, he also studied in Max Planck, and now he's in the Universidad de Guadalajara. Edgardo Álvarez Sepúlveda, he did it on development biology, and he now works in CICESE in the north of Mexico, in Baja California. And Juan Manuel Mecinas, that he did a comparative public law and international law study at Max Planck, and he now works at CIDE. So the idea, of course, is not only with Mexicans trained by Max Planck. The idea of Mexico is to return, to give opportunities to all these people that is being trained outside. But we are very pleased to say that 4 of them are already here, and we are looking to get more of them to come back to Mexico, because the idea of using the training in the Max Planck system is to train researchers to come back and to be able to give back to Mexico what they have received from this country, and also to increase science, to develop science in our country. Okay, so this is what we have about Cátedras, and we also have a call with DFG, which I am not able to pronounce in German, so I always call it DFG. And in this call that was open, we have an agreement from 2014 to 2018. We had a call on September 2016, and just to show you which were the areas, it was very open in subjects, but you can see which are the subjects that are most relevant in our collaboration. Biology was in first place, but it's also physics, chemistry, humanities, and geoscience. And we had the results on November 2017. Okay, so there are the amount of money that we are putting in these research projects, and this is a funding for 3 years. It's a very good call for research projects. Here are the names of the projects. Of course, I will not read them for you, but so that you can see that we are covering very different projects, and of course, there is a group in Mexico collaborating with a group in Germany to address the project. And this is something that I was commenting with Andreas just a moment ago, that what we have liked very much of this meeting is this collaboration. I mean, it's really one-to-one collaboration, groups from one country and the other one working together for the same goal. And we also have a very successful program that is done with the DFG, and that is used by the Colegio de Mexico. It's called Between Spaces, Movements, Actors, and Representations of the Globalization. It's like a seminar. It has been extremely successful. It has had 189 participants, and as you can see there, many graduate Mexican students and many German graduate students have been in this exchange program, coming to Mexico, the Germans going, the Mexicans to Germany, and this is we are going to start the new stage of it. These are the fields of knowledge that are used in this space, between spaces, and this is happening with the German institutions associated that you can see in the right-hand side. This is a program really on social science and humanities. I'm just giving you some of the examples which are related to the scientific development direction. There are other projects that are handled by other of the departments of CONACYT. I am just addressing the ones on scientific development, and probably Andreas will mention something about new plans that we want to develop. It's just to give you some examples and to put names on the examples, to show you that there are already postgraduate Mexican students doing a postdoc in Germany, that we have brought back some postdoctorate students from Germany, and that we have different possibilities of collaboration that we certainly invite you to use. Okay, let me just, I am in the thank you slide, so I would like to give you some data that I think is important. So we have had, as a whole, as attendees, we have had 250 people, but we have been followed by streaming by 874 people. And I want to use the opportunity to thank the Forum for Scientific and Technological, nuestro foro de colaboración científica y tecnológica, because they are the ones who are supporting the streaming for this meeting. And also, let me tell you that everything has been filmed, and you can see it in YouTube. We are very happy with this electronic complement, because we know that a lot of people follow this from even from out of the city, and that staying in YouTube, you can also watch it later on. So this is something that I wanted to mention, and to thank the forum for the support on the streaming. And I also would like to use this opportunity, if you could join me, to thank the group in CONACYT that has been working very hard to make this possible. I don't know if they are all here, but it's Daphne, Alejandro, Diana, Gloria, Montserrat, Ariadna, Paulina. Are you around? Well, some of them are around. If you could please. Thank you so much. You've done a wonderful job. And I would like to give a special thanks to Andreas Trepte, who has been the engine of all these projects. He has been really looking into every little detail. And let me tell you something that for us is a very big compliment. Andreas has become partly Latin American. Thank you so much. Okay, so now it's your turn.
So let's wait to my presentation. I would like to, let's say, to put into context this symposium, because this is, let's say, it's a photo in a long-term evolution. And there is a potential for cooperation, but we need to raise it. And you will see in a few minutes, if my presentation is coming, that we started in Latin America and other countries. So the cooperation with Mexico is in a different stage than, for example, with Argentina, where I'm living. And technically, I'm depending. ¿Qué pasa? Necesito mi presentación, hombre. Se habla español en la oficina de enlace. O sea, we are based in Latin America, but in Argentina, but I'm traveling half a year to the different countries. And my intention with the upcoming presentation is to show what could come next. Because the symposium is kind of an offer, is a kind of an exchange. I think everybody have registered that we appreciate Mexican science and you appreciate our science and you are hearing what our scientists are doing. This is the same manner like we do this kind of symposia all over the world. The last one in 2015 was in Chile. 2016 we made the same thing in Buenos Aires, and in November I will organize jointly with the FAPEST Foundation the equal meeting, different people in Sao Paulo. So, my intention is first to say who is not cooperating internationally is losing in science. And if you look, the Max Planck Society seems to be big. We are big in comparison to all institutions in Latin America. We are producing more than several nations, if you look at the number of publications. But if you look inside of our institutes, even if you take all these 23,000 people, more than 50% are not Germans. Imagine a Mexican university, CONACYT, with staff, 50% from Europe, from other countries. This is a different culture. So talking about internationalization for us means to be very international at home, in the institutes, even the directors. 35% of our directors are not speaking German or have a different passport than we. And it means also internationalization abroad. So here you see the presence of huge projects. Most are called Max Planck centers. This technique. And there is a lot of room, let's say, going south. We are near to Mexico, because we have a Max Planck Institute in Florida. This could also be a point of exchange. This is in neuroscience. They would be very interested to be a hub between doing science at the Max Planck Institute and, for example, Mexican scientists, because you are very near to them. But as you see, we have several, let's say, research facilities, because we are maintaining research facilities, and we, in a difference to, let's say, CONACYT or CONACYT in Chile, CONACYT in Chile or CONACYT in Argentina, we are funding institutes. We are not funding projects from others, so it makes no sense to ask for money to the Max Planck Society. But ask for cooperation with institutes is a very important way to do. So looking what international cooperation for us means, here you see, starting, of course, we need a legal basis, especially we learned this in Latin America. We could come to programs, we are talking now, CONACYT and Max Planck, also about a program for doctoral students. But the systems are very different, so we have to find a compatibility. But going forward, we have, of course, in projects where Max Planck institutes and local researchers are collaborating, but this is not sustainable. These are, let's say, person-to-person, face-to-face cooperations. There is nothing institutionalized. In countries like in Argentina, we are now in a phase that every 3 years we have joint calls for projects. We would like to have this also with CONACYT in the future, because this helps to establish bridges and not only personal relationships. And we have a series of different research groups which are associated to different Max Planck institutes. So if you look, what are the instruments and what could be done to improve, to develop the cooperation with us? We would like to receive more doctoral students. And we invite, because we are doing open calls for our research schools in the different institutes. My office is publishing, you will see the website, because we are maintaining a Spanish website where we are publishing all open calls every month at different Max Planck institutes. So, as I say, in every Latin American country, please feel invited. This is open for the world to send applications. But what we at the moment, what many institutes are receiving are many applications from Asia, from Europe, and a very small number from countries like Mexico. So this ball, or this, la pelota está en el campo de México, de América Latina. Postdoctoral. This is also important because we have programs to, you will see it, to support postdocs when they decide to come back, to go back to their country, to support them, and to establish something like a partner group. And as you see, we have at the moment registered 150 projects with Latin America between all 84 institutes. But we maintain more than 4,000 globally. So it's not so much like it seems. And we have 580 people working at this moment in Max Planck institutes from Latin America. But from the world, we have 8,000. So there is a lot of room for improvement. As I said, it would be wonderful to have joint calls because the German DFG is supporting projects with universities. And the DRD is funding fellowships in universities in Germany. But we are also looking and we are making, like I said, calls for fellowships, calls for projects. The other one, if you look into the folder outside this folletto, sobre the cooperation between Max Planck and Latin America, you have seen there is no page for Mexico because we are in a stage to develop tools and instruments and ways to come, for example, I hope that in this year we will see the first Max Planck partner group in Mexico. Because in this combination with the Cátedras program, there are some candidates and they are now developing their applications. And of course, we could also come, maybe in the future, when CONACYT will double the budget, yeah? To tandem groups, like we have now in Chile, in Colombia, and starting this year, we have 4 new tandem groups financed by the FAPESP in Sao Paulo. So here you see how intense in numbers is the cooperation. But of course, this is not saying so much because a country like Brazil with 140 people in our institutes, this is nothing special if you have 220 million people. But it is special if a country like Chile with 18 million inhabitants have 55 working as doctoral students, postdocs in the institutes. So, only to show you what could happen in the future, because we are giving people which are collaborating with us as heads of this different kind of independent research groups, we are giving them visibility. So in Argentina, at the moment, 5 groups. We have in Argentina also associated laboratories. It is financed by Argentina, but it is working in cooperation with the Max Planck Institute in Göttingen, and it is a huge facility. And my office is located in the Polo Científico in Buenos Aires, where Argentina is maintaining a so-called partner institute with the Max Planck Society. This is financed from Argentina, but it is working with the governments, with the principles and values of the Max Planck Institute. And so on. Only to show you with the speed of Mexican cables, the different groups in Brazil, in Chile, and as I said, I would like to see in the future more from Mexico. I would like to mention, we are participating in more facilities. We are part in project or with instruments in a lot of, for example, telescopes in Chile, but we are maintaining also instruments, and as you know and as you heard, we are doing this with this open sky policy. So these instruments are also open for cooperation within Latin America. And Julia mentioned HAWK. HAWK is, of course, a very important project where the Max Planck Institute for Nuclear Physics is cooperating. We hope that also in the HAWK South, which will probably constructed in Chile, we could cooperate. So, please follow us on Facebook. There is an official page where we are publishing news, calls, where Mexican young scientists could apply. And you could also look at this new web page of my office. So if you would like to cooperate, for example, with these new established research groups associated to Max Planck Institutes. For example, we are now organizing the first meeting in plant sciences because we have now more than 10 different Max Planck partner groups and tandem groups in 5 different countries. It will be in Santa Fe in November. And those which are looking for contacts because they never have been in a Max Planck Institute, I always say, please use the databases which are available, Scopus, Web of Science, and look what we are publishing. Because we are a very fast changing entity. So you have to look into the publications, the recent publications, because most of the researchers in our institutes, this is the bad news, don't have permanent positions. So they stay there for 2, 3, maximum 5 years. So if you find a paper 5 years ago, it is not sure that you will identify this person, for example, in this Max Planck Institute. And so fast are also changing many research topics. But by this way, you could identify partners, and please don't be shy like many other Latin Americans. Please contact these people. Others are doing this, and I'm wondering, and after 4 years living in Latin America, I think I have the permission to say this. We are missing more initiative by young people applying in our programs, because this is really an advantage to pass through one of our institutes. So with this, I would like to thank you. My contact data is on the folder outside. So who needs something, advice, help, please write us, and let's stay in contact. Thank you very much for your attention.
So it's lunchtime. thank you so much to all of you, and thank you very much for all the Germans that have come to our country. We are very happy to have you here. So let's have lunch together. Okay, Diana is asking me to ask you if you would like to have any more information from what you have heard. If there is something you want to ask Andreas or to ask to me directly now before we leave, if there is something you would like to ask. Or we have been so clear that there is no need to ask anything. I think we have been very clear. Okay, so lunchtime.